So here begins my presentation of porn. But first I'm going to start with just an overview of proteins. Proteins come in a diverse array of functions and structures. There are so many different proteins that have so many different functions. Some function as hormones, neurotransmitters, transport proteins, structural proteins, etc. But all proteins start as amino acids bonded together by peptide bonds. And there are 4 levels of protein structure. First is primary structure, which is simply the amino acid sequence that makes up the protein. The secondary structure is the conformation of the amino acids, basically how they're together, how they folded and coiled, etc. Tertiary structure is the actual 3-dimensional shape that the proteins form, and the shape is held together by hydrogen bonds, ionic bonds, when the amino acids side chains are close to each other. And then the 4th level is quaternary structure, which is multiple polypeptides that come together to form a functioning protein. So, a little background. Porn was discovered by Hashiro Nikaido in 1976. And he was nicknamed the pornologist because he studied porn, he discovered it, and he's written multiple articles about it, over 70, I believe. So, location. Porn is located in the outer membrane of the bacteria, and is also found in mitochondria and chloroplasts. As you can see, here is the outer membrane, and here's the porn. It's a transmembrane protein, so it goes through the entire membrane into the periplasm. Porn functions as a transport protein. It is embedded in the phospholipid bilayer, like I said, and allows for the diffusion of small hydrophilic molecules through the membrane into the periplasm space. It can also regulate the internal environment because it controls what is allowed into the periplasm. Now, the primary structure, there are many different types of porins. As I said, they're found in mitochondria, gram-negative bacteria, so there are different types of the porin. Essentially, they vary in the number of amino acids. However, they're about 300 to 420 amino acids long, with 274 solvent molecules, 3 calcium ions, 3 detergent molecules, and an unknown ligand. Its secondary structure is anti-parallel beta-pleated sheets. So, as you can see, they go in an anti-parallel form and loop back over each other. And porn was the first transmembrane protein for which beta structure was established unequivocally, meaning it was the first protein where it was just known that it was the beta structure. And the tertiary structure is a beta barrel. So here all the sheets come together, they coil around each other to create a barrel type shape with a pore in the middle of it that allows for things to diffuse through. Its quaternary structure is when 3 polypeptides molecules of porn linked together. So here's the 3 different porins. There's a pore right here, a pore right here, and a pore right here, but there's also a central pore in the middle that allows for things to come through as well. So the outside is hydrophobic and the inside is hydrophilic, which is unusual because usually hydrophobic likes to stay inside, whereas hydrophilic is outside. And this is because the hydrophobic outside attaches to the lipids of the phospholipid bilayer. So porn can be classified in 2 different ways. The number of strands it has, which is usually between 16 and 18 strands, and also staggered, which is simply how the edges of the cylindrical portions are held together. For example, right here, these, the edges of the cylindrical portions are held together. In conclusion, porins are very essential to whatever organism is part of. It's what allows things to come in and out of the cell without using energy, with just using passive transportation, and it also has a very complicated structure.